Welcome back to The Explainer. Today, we're diving straight into a high-stakes forensic investigation. We're stepping into the shoes of a specialized medical detective to uncover the hidden, rigorous scientific process that actually keeps our food supply safe. It is literally a world of microscopic threats, surgical precision, and split-second decisions. Let's get right into it.
Think about it for a second. Before any meat ever reaches your kitchen counter, it starts out on a farm, right? And that is an environment that's just teeming with invisible, microscopic dangers. So how do we actually guarantee that what ends up on your plate is completely safe? Well, the answer lies with the inspector. They are the ultimate frontline defense, standing right between a biological hazard and your dinner table.
Section 1. The Food Safety Shield.
Now, to really grasp what these inspectors are up against, we have to look at the massive scale of the biological threat. The numbers here are honestly astronomical. We're looking at the primary threat vectors on a live animal. Animal hides and feathers, they can carry up to 3 million bacteria per single square centimeter. And the gastrointestinal tract, get this, up to 90 million bacteria per gram of contents. That is an unbelievable amount of natural initial hazard that our food safety shield absolutely has to contain.
So, look at how the HACCP concept works here. That stands for Hazard Analysis and Critical Points. You've got those massive contamination sources on the left, but they are forced through this tight bottleneck right in the center, the physical inspection. This is it. This is the ultimate critical control point. A flawless physical inspection makes sure viscera aren't accidentally punctured. It instantly separates the contaminated areas from the sterile meat, which effectively stops severe hazards like salmonella and listeria literally dead in their tracks.
Section 2. The Inspection Timeline.
You know, this safety shield isn't just someone taking a quick glance as things move down a conveyor belt. It's actually a highly structured, chronological two-step process. We break this down into the antemortem, which is the live screen, and the postmortem, the anatomical screen. The antemortem happens within 24 hours of slaughter. It's a live observation where they're looking for abnormal breathing, maybe altered behavior, or even just an unusual posture. We then compare that to the postmortem, which is a strictly scientific, deep-dive evaluation of internal pathological lesions.
Check out the flow here from left to right. If an inspector during that live screen spots what we call a downer animal or notices it's struggling to breathe, that acts as an immediate trigger. It flags the animal, prevents killing floor contamination, and ensures humane emergency handling. And simultaneously, it initiates these highly detailed targeted forensic actions right over there on the diagnostic bench on the right.
Section 3. The Anatomical Journey.
Let's zoom in on that diagnostic bench and see exactly how a post-mortem inspection goes down, starting with poultry. Guys, this is a literal surgical procedure. In Phase 1, preparation means applying formalin to halt autolysis, that's tissue breakdown. Then, Phase 2, the inspector makes an exposure incision right at the thigh body wall and dislocates the joints to lay the carcass perfectly flat. Finally, Phase 3 is the colomic opening. They incise the bird and reflect the breast musculature forward. Every single step here is meticulously designed to expose the underlying tissue without ruining any of the internal forensic evidence.
The absolutely crucial point here is precision, because the stakes are incredibly high. Take a look at this procedural alert for gallbladder extraction. You kind of have to treat this like a biological bomb defusal warning. Bile actually has potent antibacterial properties. So if an inspector's knife slips and ruptures that gallbladder, the spillage into the cavity will completely destroy the integrity of the sample. It ruins any subsequent bacterial culture results. It's just game over for that test.
Now, if that extraction is clean, the inspector moves on to targeted checks. Here we see two really distinct focus areas. On the left, they're swabbing the tracheus mucosa to check for things like avian influenza or Newcastle disease. Then on the right, we're seeing the shocking visual evidence of a systemic issue. This is the infectious bronchitis virus, specifically the QX variant. This pathology is unmistakable. It actually prevents normal shell formation, which results in these bizarre shellless or soft-shelled eggs, along with severely atrophied oviducts.
They also rely heavily on precise diagnostic cheat sheets, kind of like this anatomical map for poultry coccidiosis. By mapping exactly where the lesions are located in the intestines, inspectors can trace specific parasitic strains. For example, if they open the bird and find the KK totally packed with highly pathogenic necrotic cores, they instantly know they're dealing with E. tenella, which is a nasty strain that causes huge mortality rates in young birds.
Section 4. Rules of Carcass Judgment.
So finding the clues is really only half the battle. Now we transition to the decisive moment. How do inspectors synthesize all this evidence to make that final pass or condemn ruling? Well, the rules for judgment come down to this specific matrix. It plots the time frame, acute versus chronic, against the spread, localized versus generalized. A localized chronic issue, like a contained abscess, might just be trimmed away, allowing the rest of the meat to safely pass. But look down at the bottom left. Rapid, acute, systemic spread, showing multiple inflamed lymph nodes. That demands total carcass condemnation. Other conditions warrant an automatic total rejection too, like a fever or pregnancy toxemia, which inspectors can actually identify just by a distinct ketone smell.
And how exactly do they prove systemic spread? By hunting down the lymphatic system. Look at this protocol for cattle. If a generalized disease like tuberculosis is suspected, the inspector doesn't just glance at the surface. Oh no, they dive deep into specific lymphatic clusters, the prescapular nodes in the forequarter, the precrural nodes in the hindquarter, and deep visceral clusters. Any evidence of lesions across these nodes immediately dictates total condemnation.
Section 5. Mammals and Special Cases.
All right, let's expand the scope a bit. These rigid rules actually have to adapt because animal anatomies are vastly different and honestly, sometimes full of surprises. Notice how the surgical requirements build across different mammals. For cattle, inspectors have to make deep incisions into the outer mastication muscles and the heart septum just to find specific tapeworm cysts, like Cysticercus bovis. And for horses, in endemic areas, the inspector is actually required to split the head lengthwise to examine the nasal septum for a disease called glanders. Crazy, right?
But here is a truly fascinating curveball for you. If this entire systemic evaluation process relies on tracking infection through the lymphatic system, how in the world do you trace and stop an infection in an animal that has absolutely no organized lymph nodes at all?
Enter the ostrich. Yep, ostriches lack an organized lymphatic system. And because of this, viral and bacterial infections aren't easily localized. They generalize incredibly rapidly throughout the entire bird, so inspectors have to completely pivot their strategy. Instead of checking lymph nodes, which don't exist, they rely on key anatomical checks, examining the heart and the lungs directly for things like hemorrhages and pneumonia.
And then you have wild game animals, like kudu or antelope, which present totally different clues. Their herd health is usually excellent, so you rarely see systemic pathology. Instead, inspectors are looking for management risks, like heat spoilage or gunshot trauma. They also run into what are called aesthetic parasites, like stylisia in the liver or cordophilus encysted in the heart. These look super alarming, but structurally, they're generally not infective to humans. The action here is simple: trim the affected areas and safely pass the rest.
Section 6. The final verdict. Okay, it's time to bring all of this together and synthesize the overarching principles of this immense public health responsibility. Across all of our sources, 3 core best practices really emerge. Number 1, standardize the approach. Having consistent, chronological, antemortem and postmortem protocols is absolutely non-negotiable. 2, know the species nuance. You've got to tailor your incisions to the specific anatomy right in front of you. And 3, protect sample integrity. The procedure itself must never ever become the contaminant.
Ultimately, whether an inspector is making a deep heart cut in livestock to find a tiny cyst or racing to identify rapid viral spread in an ostrich, this structured scientific protocol is the only way to reliably prevent generalized diseases from ever entering our food chain. It is a daily, relentless application of forensic veterinary science.
So, I leave you with this. Next time you sit down for a meal, will you see the invisible shield? Will you think of the hidden scientific rigor, the deep anatomical checks, and the dedicated medical detectives who meticulously cleared that food for your plate? Thanks for joining me on this explainer, and keep asking questions about the amazing world around you.